Classification by staining type: how microbiologists see the invisible. In their natural state, most bacteria are nearly transparent, making them almost impossible to study under a standard light microscope. Staining isn't just about adding colors, it's about exploiting structural differences. By adding specific dyes, we can reveal the hidden architecture of a cell, dividing the microbial world into distinct groups based on what their cell walls are made of and how they survive. These techniques turn a blur into diagnosis. The most critical structural feature in bacterial staining is peptidoglycan. This is a rigid mesh-like polymer made of sugars and amino acids that surrounds the cell membrane. Think of it as a protective case. The thickness of this case and what sits outside of it is what dictates the result of the most famous test in microbiology, which is the Gram stain. The Gram stain is the gold standard of bacterial classification. It targets the peptidoglycan layer, a mesh-like scaffold of sugars and amino acids that provides structural support. Gram-positive bacteria have a thick, multi-layered wall of peptidoglycan that traps the primary stain, crystal violet, turning them a deep purple. In contrast, Gram-negative bacteria have a much thinner peptidoglycan layer and an additional outer membrane. They lose the purple stain during decolorization and must be visualized with a pink safranin counterstain. Whether we are looking at the thickness of a peptidoglycan wall, the waxiness of a mycolic acid cell, or the presence of a dormant spore, staining gives us a visual window into bacterial survival. By understanding these chemical differences, clinicians can make faster, more accurate decisions that save lives. But what if the cell wall is literally too waxy for water-based stains to penetrate? This is the case for the genus Microbacterium, which includes the pathogens responsible for tuberculosis and leprosy. These bacteria possess mycolic acid, a long-chain fatty acid that makes the cell wall extremely hydrophobic and resistant to traditional Gram staining. To identify them, we have to use the acid-fast technique, which uses specialized methods to force dyes past that waxy barrier. The acid-fast process starts with carbon fusion, a bright red dye that is driven into the waxy wall using heat or detergent. Once the dye is inside, it stays there. Even a harsh acid alcohol decolorizer cannot strip the red from these acid-fast cells. For everything else in the sample, the acid alcohol washes the red away, allowing them to take up a blue methionine counterstain. The result is a striking contrast: red acid fast rods against a sea of blue non-acid fast materials. Finally, we have the Endospore Stain, used to detect some of the most resilient structures in nature. Genera like Bacillus and Clostridium can produce endospores, dormant, highly resistant structure that protect the bacterial genome from heat, radiation, and chemicals. Because spores are so impermeable, we must force malachine green into them using steam. We then wash the slide with water, which decolorizes the normal vegetative cells but leaves the spores green. A safranin counterstain then colors the vegetative cells red, making the green spores stand out clearly. To summarize, Gram stain differentiates bacteria based on peptidoglycan thickness, resulting in purple or pink cells. Acid-fast stain identifies bacteria containing mycolic acid, appearing red, while non-acid fast cells appear blue. Endospore Stain highlights bacterial spores in green and vegetative cells in red. Each staining technique serves a unique purpose in bacterial identification. Keep sharing knowledge and continue exploring the fascinating world of science with me. See you in next video. Bye bye.